organelle is kind of like the postal service. What it does, it packages substances that then the cell exports. The packaging material itself is actually cell membrane, which is really great because when we export the stuff, it fuses with the cell's own membrane as the exportable substances are expelled, which is even greater because we replace the cell membrane all the time. And I forgot to mention the organelles, especially the ones like an ER, like here, that have membrane around them. That's the same cell membrane, it's phospholipid bilayer. We use this phospholipid bilayer all over the place. Because if we can use a thing and we use it over and over for many, many different things, it's great for us in the body to use. It's very efficient that way. So that's, what is this organelle called? Golgi Apparatus. So that's the packaging plant. The vesicle membranes. Vesicle is that little thing that is the packaging material comes in a vesicle. So it's the packaging itself, it's the vesicle, and that cell membrane, and then when it expels whatever on the inside, that cell membrane gets part of the regular cell membrane of the cell, and it's a renewal process that way. So that's pretty cool. Just a few facts to help. Well, that's a good one. When we get stuff into the cell, it's not all bad. Some of it is bad, but we still have to dismantle it and check out what's going on and take it apart and maybe destroy it. And so that we have an organelle that does that because it has these digestive enzymes in it. What organelle is that? Lysosome. It's one of my favorites. Lysosome. It can be used to do what they call autolysis. That means auto means self. So cells do self-destruct. So if you are feeling sorry for yourself, you have cells in your body, they die for you. Just know that. I know. When you ever have pus, you know those are actually cells that die for you. It's not just gross.
So would it do it when it was not functioning correctly, or
yeah, or when it's got a host, and it says, hey, you know, you got to kill me because otherwise it starts getting way worse for the whole body. So you have that as well happening. Some of these cells, they take up a bad thing, a bacteria that they don't want, and then they actually tag themselves as, hello, I've been attacked, and I got myself like calyx on me too. So the immune cells go, oh, we got to do something about this. And so then yeah, it's a molecular alarm, absolutely, because then what happens after that process, that often stimulates the specific immunity. So then from that sort of, this cell ate this thing and figured out something's not good, then the BT cells can go in and go like, okay, we got to go for it. And so then that process can be initiated. So it's pretty nifty.
When you have an autoimmune, are the lysosomes attacking like everything that's coming in? Or?
No, the autoimmune pathologies are more specifically that autoimmune cells attacking your own cells.
What are those cells?
Well, there will be immune cells, the T cells, B cells.
T cells, B cells?
Yeah, this is inside every cell where you have lysosomes, and it's a lot of chemicals. If you're looking at a T cell, they actually have something called perforins, which are really bullet-looking proteins that they shoot out of themselves, kind of sort of out, penetrate the other cell membrane, and then you know, it's like in a body, when you make a hole in the body, it's going to bleed out, and so that causes the cell to die as well. That's more like that process. Another thing we have in the cells, I didn't have a question on that, these are the centrioles. And these are kind of interesting things. When we look on the inside of the cell, let me go back to a slide that I skipped. Where did it go? Here. When we look on the inside of the cell, we actually have a lot of microtubules and scaffolding things that do both. They create shade for the cell, they hold the cell up, make sure it doesn't collapse depending on where it's at, but they also create pathways for molecules to walk on and to carry things to the cell. So we call that collectively the cytoskeleton that does that. And when we go back to this place of the centrioles, that's sort of that kind of material. So these sort of like combination or system of these hollow tubes that look very specifically, they look very symmetrical and pretty, actually. But what they do is they help in the cell mitosis, which is the cell division. And so when we want to have a cell that divides, this is basically here. So it looks weird because this is a cell here, right? I'm trying to draw a cell membrane around it, and it's a cell that's going to divide. But you see these little things on both sides, on both poles. These are these centrioles. There's two of them in each cell. And what happens when the cell divides? You have to copy the nucleus, the DNA material, because every cell will have to have a full set of genetic material. Well, most cells, but that's the fundamental, so we can then cause a division. And we will then have a moment where we need to figure out how can we have half of the copy go to one cell and half of the copy go to the other cell. It's not scrambled. And that process is really done by the centrioles. Because as they go to each corner type of the cell, they can then have the DNA material, which is this blue stuff here, is the DNA material right here. They can pull half towards one side and the other half towards the other side. And that assures that the genetic material is represented fully in each daughter cell. And so that's kind of a very specific organelle, but of course very, very crucial when it comes to cell division. Does that make sense a little bit?
Is it contracted for the DNA material?
Well, they create these arrays, these asters, these tubes. There's actually microtubules. The video that I have on this cell thing is really cool about that. If you watch that TED Talk, I'll show you at the end which one I mean. And they actually, these molecules, then they walk. There's molecules that walk the DNA down to each side of the pole. And so my thinking is, not being a cytobiologist kind of guy, but my thinking is these create the scaffolding, the framework for these rays to go across, and then go from each side to the center of where these DNA molecules are located, and then they can walk towards them or they pull them towards them. But I think from what I know and what I've learned, especially this process, it's pretty physical. To the smallest degree, it's molecules walking. It's crazy. So, and for that matter, maybe we don't even know the whole thing anyway. Still, maybe we're just kind of guessing. But that's partly why there is part of biology that really tries to visualize what's going on, to see it, because we have a better way to understand than we can see somehow. I still think feeling is way better than seeing, but that's just because I kind of no matter, let's go to the next slide. No matter what the cell's job is, any work it does requires energy. What is our energy molecule? ATP. Who makes the most ATP in the cell? Mitochondria. There you go.
What was the first? What is the first thing?
The first question was about the ATP. I want to make sure you guys know ATP is the energy molecule.
Oh, the molecule.
Yeah. Yes?
Before the mitochondria was introduced into the cell, how did it obtain ATP?
Well, if we think about how it does obtain ATP without the mitochondria, it's basically called glycolysis or glycolysis. Knowing the word lysis, you know now when it says glucogen and lysis, it breaks apart the glucose. So you can extrapolate. A lot of what we're doing in this kind of science is you take the words apart and then they make much more sense. So never study too fast. It's very important to be slow in this kind of stuff. And so glycolysis or glycolysis breaks it down, glucose to pyruvic acid, and that releases a few ATPs. But it's nothing. I think it's 2 net ATPs, I always forget. But it's nothing as in the mitochondria, because in there you make like 36 net ATPs. And so I think the mitochondria came in a long time ago into these cells. But it has its own DNA, which is interesting because that means at some point the mitochondria was its own structure. It's its own bacteria. So that's another place where we can see we're actually really, I'm helping my kid with microbiology a little bit, and it's interesting because I haven't had that in so long. But you know, we're just part of that whole microbiome. We just don't have to get too cocky about it. Be humble that we're and grateful that we're actually alive on this planet. But this here now brings me to the nucleus, which is a large sphere in the center of most cells. It holds and protects the instructions of how to make all the different types of protein in the body. Most fundamentally, this cookbook is made up of 4 randomly repeating molecules called nucleotides that form a strand. Each strand has a copy. 2 of them make up a double helix. The sequence of those 4 molecules determines the sequence of amino acids that make a protein. Do you get that? How that nucleotide after nucleotide after nucleotide in that double helix is sequenced. One comes after another. It's very simple, very fundamental. That determines what amino acids makes up a protein. So we're going to have this thing where we have this thing where 3 of these things will make 1 amino acid. And so we have to go from nucleotide to amino acid. And that's a central piece in biology that we'll have to do. But before we do that, some cells don't have a nucleus. And when the cells don't have a nucleus, they're unable to divide. And there is one example in the body that is most famous for that. What is that? The red blood cell. So the red blood cells were one example that doesn't have a nucleus. And since there's 25, whatever, many of them, that is pretty important. There are a lot of them. All right, nucleus, dark sphere, housing the genes. We can also, the genes are hereditary characteristics, the things we inherit. Most cells have 1, RBCs has none, and some have multiple. It's also interesting. A muscle cell has multiple nuclei in it. And the reason for that is, when you look at a muscle, you got one muscle that goes all the way from here to here. It's a long, long muscle. The whole length is 1 cell. They're not multiple cells one after another. And so when we're made, they are 1 cell after the other because it's a long, long, long cell. It's a thread. And so in the making, those small little pieces, they fuse together and make 1 long thread. And so the nucleus stays behind, and so there's multiple nuclei in that cell. But that's very specific. On the inside, 2 elementary membranes filled with pores create the boundary from the nucleus to the cytoplasm. That's right here. But then the other thing is, we also have an area on the inside that's known as the nucleolus. That is like a separate place inside the nucleus. And that is a place where we make ribosomal RNA. When we get to RNA, we'll have multiple different RNA. For us, most of the time, RNA is a copy of the DNA that we want to make a protein with. But the ribosome itself is part RNA. So now the question will arise, how are we going to be able to make a protein with the information from the nucleus without making the ribosome? We have to somehow make the ribosome to start that process. And that's why we're going to have a nucleolus that can create those ribosomes, those mRNAs, which are part of the ribosome. So we think of that as a ribosome kind of. And the nucleolus has to make that, then the cell can actually make proteins from its DNA that's in the molecule. So that's what the nucleolus does. It's a little bit confusing. It took me a few years to get it. If you get it in one sitting, you're pretty darn smart.